in testing carcinogens for DNA damaging properties, working with industry, regulatory bodies, and international collaborators, including VIT. His group is focused on in vitro approaches to replace animal testing for carcinogens. His cancer research is focused on early diagnosis of esophageal cancer and is currently assessing blood-based mutational diagnostic approaches to identify high-risk cancer patients and address questions about exposures to carcinogens. He's a highly reputed established researcher with publications of 263 and H index 31. He's presently the president of United Kingdom Environmental Mutagen Society. Professor Gareth, pleasure to see you virtually today, again this 2020. Now I welcome Professor Gareth to present his talk on Swansea University Medical School helping to supply the drug pipeline.
So, thank you and welcome. So, can everyone see my screen? Is that working okay?
Yeah.
Okay, so thanks very much for the invite to come and talk at this Indo-UK virtual conference. It's a great pleasure to be here. So, as the chairperson just mentioned, Professor Sarawathy, so sorry for mispronunciation. I'm going to talk about the drug pipeline and how Swansea Medical School are aiding in developing and continuing that. So, for those of you who don't know Swansea University, I thought I would just position us geographically. So hopefully you can see the pointer on the screen. So Swansea as a city is based here. So this yellow area on the UK map is Wales, and Swansea is 3 hours away from London that you will all recognise. So we are based in a different country as part of the UK. So Wales is a separate country of the UK, and we are lucky enough that Swansea is based on what is known as the Gower Peninsula. Here's a finger of land that sticks out into the sea that was the UK's first outstanding area of natural beauty. So it is a really spectacular place to live. So this is an image of the two campuses of Swansea University. The university was founded in 1920. And so you will see this logo appearing lots of places because we are celebrating our centenary this year in 2020. Unfortunately, 2020 hasn't been the year that perhaps we wanted it to be, and our celebrations of our centenary have been cut short by COVID-19. Swansea University has the distinct honour of being the closest UK university to the beach. You can see both the campuses here are right on the seafront. And we are home to about 20,000 students, so we're a middle-sized university in the UK with about 3,500 international staff. The important thing I wanted to point out is this group of buildings here on the top map, on the right-hand side of the screen, are what is known as Singleton Hospital. So the medical school is located on the campus immediately adjacent to one of the two main hospitals in the city of Swansea, which is really important for us as a medical school because that allows really close clinical collaboration with the doctors and other healthcare workers, and also gives us access to patients and patient samples much more easily. So Swansea University is pretty well regarded in the UK for its teaching and for student satisfaction. Lots of students enjoy coming to Swansea University, and there have been occasional awards that the university have won in the UK university context. So the university is based around Swansea City, and the city of Swansea is a vibrant area, so seafront location, And the students like coming to Swansea University as well, partly because it isn't a really expensive part of the UK to live, unlike the major cities like London. So the cost of living makes it affordable for students to come here. But the main reason, as I mentioned earlier, that both students and staff are attracted to Swansea is the outstanding areas that Swansea is based in. So the Gower Peninsula is home to a number of outstanding beaches. This one here, there's a group of students walking along Rosilly Beach with Worm's Head in the background, this rock-like structure. And this was voted in 2017 as the best beach in Europe. So the students really like their downtime, and they are very attracted to coming to work at a very good university, which is in an outstanding area of natural beauty. So obviously the university is made up of many colleges, and the medical school is one of these entities. And we work together very closely. We have lots of collaboration, multidisciplinary collaboration across these different parts of the university. But we have a really close working relationship with the local health boards that cover this part of Wales. So in the UK, the NHS, the National Health Service, is devolved into regional health boards which run the health services, and there are two based in this part of West Wales, which we have formed a partnership with. The university and these two health boards have formed a partnership to work more closely together for both research purposes and teaching and learning purposes as well. So the university are able to work closely with the NHS to train exactly the right kind of healthcare worker of the future, and by working together it enhances our capacity for doing research and getting large number of patients into clinical trials and other things. So it's a very key collaboration, and the collaboration is called ARCH, a Regional Collaboration for Health. We also work very closely with lots of industrial companies. So Swansea University, founded in 1920 by industry. So Swansea University was founded by the large manufacturing industries, the heavy metal industry, the heavy industries of coal and steel and other metal industries. They wanted a university to train their staff to maximize their industry, and Swansea University was the result. And to this day, Swansea University works closely keeps that industrial heritage and works very closely with companies. So these are some of the companies that the medical school work with. If you looked at other schools and colleges in the university, they would be working with all sorts of other companies as well. Some of these are very large industrial pharmaceutical companies, and many of them are small, vibrant enterprises which are developing in South Wales around the healthcare sector. So the medical school. So I asked a colleague of mine for a few slides about the medical school, and the first one that I was given says, is Swansea University Medical School any good? Well, I suppose I'm biased because I would say yes, obviously, but I think that there are pretty good indicators that the medical school is held in reasonably high regard. So in the UK, all universities enter into this competition every 7 years called REF, the Research Excellence Framework, and this ranks subjects across the whole of the UK for research quality. And in 2014, the last time we had one of these, the medical school came second out of the 95 competing universities that was submitted. And we came joint first for an element of this called research environment. And in research environment means the facilities and the attitudes and culture that we develop our research, our researchers. So the way that we treat staff and students to foster a friendly and healthy research environment. So the medical school is pretty good. So what do we do? So I should say that what I'm doing at the moment is going to give you an overview of the medical school, give you a real sort of whistle-stop tour of the things that we focus on in terms of research. It is impossible to do a really thorough job of this in a short seminar to you today, because we have 150 or so academic staff and 200 to 300 research staff working on a really diverse range of different research topics. So I'm going to pick out a few highlights to focus on today, to give you an overview of the work going on in the medical school. And then I'm going to spend the second half of my talk focusing on my research. So I'm really passionate about research, and given any opportunity, I will talk about the research that I'm really excited about. And so I'll talk to you about the research that my group are interested in studying DNA damage and mutations. But before that, I'm going to just give you an overview of the medical school, just so you can see the kind of background in which my group work. So in the medical school, we have 4 themes of research. So the first one is around biomarkers, medical devices, microbes, and patient population. This is health informatics mainly. So these are the 4 themes that we research in the medical school. So each of these themes has a large group of academics and researchers working within them, and they're coordinated by a lead person who then tries to make sure that the research is coordinated, so we maximize the opportunities that we can get out of this research. And so in the context of the title and the theme of the conference about therapeutics, the title of my talk was about feeding the drugs pipeline. So I've tried to give a flavor of how we do that in terms of the medical school's wider research. So the first thing to mention is that we, in our biomarkers research theme, are researching the very heart of different disease states. So we're looking at the nitty-gritty, the real detailed genes and proteins which drive certain types of disease. And we do this for dual purposes. So we are looking for those key signature events that occur which might become biomarkers of a particular disease. But by understanding the mechanics of what's happening in that disease, they are also druggable targets as well. And so there's lots of these targets become picked up by different pharmaceutical companies or different small companies who are trying to then use them as druggable targets to create therapies to try to treat that disease. And we work across a broad range of different disease settings. So cancer is something that I am really interested in, but we have a lot of people working on neurodegenerative diseases, cardiology, diabetes, regenerative medicine, reproductive health, and many, many others that I haven't got time to talk about today. Within our microbes and immunity theme, there is quite a focus on antimicrobial compounds, as all medical schools, all research institutes are interested in this. With the advent of resistance to antibiotics, new antibiotics are needed. And we have a research group that is really interested in the actual P450 enzymes that are crucial in metabolizing key pathways, allowing microbes to grow and thrive. And this group are really interested in using those cytochrome P450 enzymes as drug targets to treat both human pathogens and to be used as pesticides in an agricultural setting by targeting fungi and other problem organisms which damage crops and livestock. In the medical devices theme, there's a lot of activity around biosensors for diagnosis. Mainly this is carried out at the nano scale. So I'll talk a little bit more about the nano health work that's carried out in a few slides' time. But a lot of focus also in this medical devices theme around nanomedicines, creating new medicines by tagging nanomaterials to a drug payload, whether that be a chemotherapeutic agent or using the specificity that the nanomaterial can provide to deliver target payloads to tissues. And then the other theme does a lot of health informatics research. This is really important for the drug pipeline because it allows clinical trial design to be tested and formulated. It allows monitoring of drug side effects during trials and after licensing of the drugs. there's a particular focus within the health informatics area around the impact on mental health, but many other diseases and situations are studied as well. And this is done at a population scale, as I'll explain a little bit later. And the other thing that the medical school are involved with in terms of developing and enhancing the drug pipeline is through actual clinical trial studies. So this picture on the right-hand side of the slide shows a picture of one of our buildings, and through this glass window at the end is actually the front. Across the road is Singleton Hospital. So the hospital is right across the road, and just to the left and right of this foyer here is what's called our clinical trials unit and our joint clinical research facility, which has both an imaging suite for imaging patients during trials. And so patients walk across the road from the hospital to come and have scans performed as part of their trials. And on the left-hand side of this foyer, there is a 14-bed clinical trial ward, which is staffed 24 hours, and actual clinical trials are carried out there in conjunction with the local hospital. So there's a lot of activity around the medical school around the drug pipeline and developing further drugs and testing their efficacy and their tolerability. So I thought what might be useful would be to take you on a little bit of a tour of some of the buildings where some of this work is carried out. Again, this is going to be a bit of a whistle-stop tour, but it may allow you to get a bit of an impression of the medical school as a whole. So this building at the top here is the Institute of Life Science 1 building, the ILS building. So the medical school in Swansea is 16 years old this year. We were founded in 2004. So that is very, very young for a medical school. We're one of the youngest medical schools in the UK. And this building was built in about 2007, 2008. So it's about 12 years old. It was the first research building that was built by the medical school. And this is where a lot of the wet lab research is carried out. So there are 5 floors of wet labs, which have state-of-the-art molecular biology equipment, facilities, and state-of-the-art cell facilities, both in culture and in imaging and other technologies to study both the molecular and cellular aspects of different diseases. The next building here is the ILS2 building, which is shown in the top picture. And so the ILS2 building was built in around 2012, so it's about 8 years old. But the lower wing, which is featured at the bottom, is actually a separate entity to the ILS2 building, and it's called the Centre for Nanohealth. And so the Centre for Nanohealth is a pretty unique interdisciplinary research centre where we are using engineering and physical science principles to answer questions of a biomedical nature. And this is a key interdisciplinary collaboration between the medical school and the colleges of both science and engineering in conjunction with the local NHS, so through Singleton Hospital. And in this facility, there are lots of different research activities taking place, which focus on things like nanomedicines, as I mentioned, and using nanoscale blood clotting through complex rheology assessments, as well as nanosensors. And the nanosensors which are being developed are detecting a range of different phenomena, including oxidative stress. And I believe that one of my colleagues, Dr. Anita Devados, is going to be talking to you about some of her really exciting research about using nanosensors to detect blood-based markers of oxidative stress and prediction of different diseases. And then the last building that I will just mention briefly is the third building that was opened for the medical school in 2015, which is the data science building. The data science building houses what's known as the SAIL data bank. So the SAIL data bank is the secure anonymized information linkage data bank. So effectively it is a data warehouse containing the health records of the whole population of Wales. So the population of Wales is 3.5 million people. On an Indian scale, that is tiny, I know, but I think on On a UK scale, that is a sizable number of people. And the scale of the Welsh population has been seen to be perfect for developing these novel linkage platforms. Some attempts to create the similar kinds of information warehouse for bigger populations have failed because they have tried to contain too many records for too many people too fast. And so the SAIL data bank, working on a population of 3.5 million, has achieved significant success, mainly because of the scale that was achievable of around 3.5 million people. So this data bank contains all of the primary care data for every patient in Wales. So this is not just a snapshot of activity. This is a historical activity. It goes back up to 20 years and is updated on a weekly basis to contain every new visit of every member of the population to a GP and the outcome in terms of what that GP visit resulted in terms of diagnosis, test results, etc. It also contains all the secondary care information from hospital visits, scans, endoscopy, tests, clinic reports from various different clinics, depending on what the patient has been in hospital for. But it also contains prescription records, education records, social care records, and many, many other records. And they're all integrated together so that they are, you're able to identify for a single patient their whole health life story at the click of a button. But far more important is the ability to combine all those patients together to create cohorts of patients with similar conditions, undergoing similar or sometimes different treatments, and then monitoring their health outcomes in real time going forward. And so this is a really important resource for health informatics research. And the picture in the slide here is of Professor Ronan Lyons, who was really the architect of using this kind of platform to drill down and mine the data for all kinds of different health outcome purposes. And he is one of the leading international lights in health informatics, and he presides over the SAIL databank at Swansea. So that is a whistle-stop tour of the medical school, and I apologise that I have covered a fraction of what we actually cover, but I wanted to give you an overview of the research that occurs in Swansea University Medical School. And I'm going to spend the rest of my talk talking about something which is close to my heart, which is my own research. And as I said earlier, I'm very passionate about research. Lockdown has been a torture for me because it has robbed me of going into the lab, and I am happiest when I am in the lab talking to the PhD students and postdocs. So I'm going to talk about my research. So my research focuses on DNA damage and the induction of DNA mutations, and I've had a 25-year interest in this area. And many of you will know that DNA damage is really important because DNA damage, so there's various types of DNA damage listed on this slide here, attacking the DNA double helix, result in DNA mutations that occur. And so DNA damage causes mutations in DNA which get fixed, they get passed down to daughter cells, and this causes cancer later on. So cancer is a disease of mutations and is driven by the accumulation of mutations. And those mutations come from the DNA damage that we acquire in everyday life. And so this is a hugely important area to study. And it's really important for the drug pipeline as well, because testing new products, so pharmaceuticals in particular, but also other products such as industrial chemicals, cosmetics, foodstuffs, and other things, testing those new compounds for their ability to cause DNA damage is vital, because we need to prevent the licensing and approval of any substance, any new substance, which causes DNA damage, because we will find out in 20 to 30 years' time, if we were to do that we would have an increase in specific cancer rates. And so to prevent that uplift in cancer development, we have rigorous tests which make sure that all products, including pharmaceuticals, do not cause DNA damage or mutation. So DNA damage is also known as genotoxicity. So geno meaning DNA, and obviously toxicity meaning damage. And so it's vital that we use DNA damage as a short-term measure of carcinogenicity, of cancer induction. We can't use cancer development itself in humans as a way to study whether a new pharmaceutical is a carcinogen or not, because there's a 30-year lag before the cancer actually appears. So DNA damage is a short-term surrogate marker for whether a compound is likely to be a carcinogen. And these short-term DNA damaging tests are used in vitro in cell models to begin with, and then they're used in vivo in some cases, usually in rodents, to confirm this in vitro data. And it's fair to say that a positive result in a genotoxicity test for a pharmaceutical or for a foodstuff or another compound pretty much terminates the development of that compound, because it is a really high risk to pursue with the development of a compound which is known to cause DNA damage. So cancer is a big problem, obviously, we all know this. So this is data from CR UK, Cancer Research UK, which shows that there are about 14 million new cancers diagnosed per year. It's probably underreporting, because there's probably a lot of cancers which go undiagnosed, particularly in developing countries. And of those 14 million new cancers, there are coupled to that, there are 8 million cancer deaths every year. So it's an enormous task to tackle this cancer burden. And it disproportionately affects developing countries over the more developed countries. But it's also true that about 50% or more of these cancers are actually preventable. So they're caused by our exposures to things in our environment, our lifestyle choices, our occupational exposures, etc. And if we could identify those more and reduce them, we could reduce half of the number of those cancers. And as an example, so worldwide, the most common type of cancer is lung cancer. And the majority of these lung cancers will be caused by cigarette smoking. And so by removing that cigarette smoking, we could hugely reduce this level of lung cancer incidence across the world. And so this is another reason why the safety testing of novel pharmaceuticals is important, because we have such a high background of cancer that we don't want to add to that by introducing new products onto the market, which are going to increase this level of cancer further. And so to detect the presence of or the induction of this DNA damage, there are a range of different tests carried out. And there are what's regarded as 3 different types of DNA damage that can occur, or DNA mutation that can occur. The first is a point mutation, where a single base gets changed for another base, so a guanine for a thymine, for example. This is a point mutation in the DNA. But you can also get DNA damage in the form of broken chromosomes, where chromosomes get snapped, so the chromosome actually detaches and a fragment free floats and disappears somewhere. You can also get chromosomal copy number changes, where you gain or lose whole chromosomes, a term called aneuploidy. And because there are 3 different types of this damage or mutation, you have to use multiple tests which are aimed at detecting each of those different types. So this is why we have a battery of in vitro tests. And these in vitro tests are carried out on new pharmaceuticals or new other entities. And it is hoped that this battery is able to detect any of those compounds which are causing DNA damage, and then that is used as in a regulatory context to decide whether to allow that compound to be licensed and marketed. It sometimes produces some ambiguous data, because you sometimes get some of those tests coming back positive and some negative, and so that can mean that interpretation of the data is complex. And it certainly is a search on for newer and better tests for DNA damage, which might be more predictive. And in a nutshell, that's one important area that my research group are interested in, is the development of better in vitro tests for DNA damage for genotoxicity. So this is a regulatory framework. I mentioned that compounds have to go through a regulatory testing approach to make sure that they have the regulators are happy with whether a compound is truly positive or negative. And this is a representation of that. So this is the suggested testing guidelines from the COM, which is the UK government's Committee on Mutagenicity. So I sat on this COM committee for 10 years and was involved in the development of these guidelines, which is why I've chosen to show these here. But essentially what this shows is that there are stage 1 tests, which are carried out in cells in culture, and stage 2 tests, which are carried out in rodents usually. And for some compounds, if they're negative results in vitro, that is sufficient to be able to say this compound isn't mutagenic, therefore it can be pushed forward for licensing and for sale. For pharmaceuticals, they tend to have to go through a stage 2 in vivo test. And there are various different results that can come out of this, either being positive, negative, or equivocal, where you can't quite make up your mind. And so there's this testing, this framework for testing to protect the human population. And so in my research group, we are really interested in designing new and more sophisticated in vitro tests for studying both genotoxicity and for studying carcinogenicity. And we also are really interested in using the same methodologies which measure DNA mutations, DNA damage, in patients. And so ask the question, can this tell us anything about diagnosing cancer patients? And I'll talk a little bit about that in a few slides' time. We are spending a lot of time at the moment working with developing three-dimensional tissue models, because the standard in vitro tests are carried out in 2D cell models, which are not really representative of the human physiology at all. And so by developing more complex 3-dimensional tissue models, and we've developed models in skin, in liver, and in lung, we hope that this will allow us to better predict whether compounds are a threat to our health or not. And a large part of this is aimed at reducing the reliance of animals, so we can move to an in vitro system primarily without having to use lots of animals, rodents mainly, in testing. So one of the tests that we use a lot is the so-called micronucleus assay. And so the micronucleus is what, as the name suggests, it is a small nucleus. So it is a mainstay of genotoxicity testing. It is used both in vitro in cell models, and it is used in vivo in rodents to assess whether a DNA damaging event has occurred. And so micronuclei are markers of chromosome damage. So in this schematic here, this cell at the top in the center has lost a whole chromosome here, or a fragment of a chromosome, which end up being micronuclei, either a whole chromosome containing one or a smaller micronucleus which contains a fragment of a chromosome. You can distinguish between these two things by staining the chromosomes for what's called the centromere, the center of the chromosome here, and so the fragments don't have that. And in this bottom picture here, you can see a fluorescently labeled centromere painted cell where the micronucleus in the middle stains with the centromeric paint, showing us a whole chromosome. You can also get this tug of war situation where the chromosome gets pulled between two nuclei, as is shown in this bottom picture at the right here. Unfortunately, this chromosome will snap eventually, and so it will cause a DNA damaging event. And so micronuclei are really useful in markers of DNA damage because they can be used in various different cell types and species. You can use them in different cell types as shown up here, and you can use light microscopy and stain them with stains like Giemsa, or you can use fluorescent stains to stain the cells and use fluorescent microscopy to look at them in more detail. So we are fortunate that we have purchased two automated systems for the micronucleus assay. So The micronucleus assay is very good at detecting genotoxic or mutagenic agents. And automation with this kind of system, this is a Zeiss Metaphor system. We have 2 of these systems. This allows us to really capture data much faster and much more powerful data as well. And so we can capture data in a couple of hours that used to take us a couple of days when we would be scoring these cells manually. We also can up the number of cells scored. So we tend to score around 10,000 cells in our micronucleus assays per dose, whereas conventionally people would maybe score only 1,000 cells per dose. And this allows us to capture really detailed dose-response relationships, just such as this one shown here, where you can clearly show threshold effects, separating a safe exposure level from an adverse exposure level. So the in vitro tests, like the micronucleus test, are well established, but they are not perfect. So all of these in vitro tests were developed with sensitivity in mind. This was part of the precautionary principle. They wanted to make the tests really sensitive. But the problem with that is that they dropped some of the specificity along the way. And so the tests are not always specific, and that means that some compounds test positive in these in vitro tests when they aren't really DNA damaging at all. And these false positives mean that we discard lots of valuable compounds. So if you think of pharmaceuticals, there are drugs in drug development which have been lost because of the misleading positive result in a safety test somewhere. This also requires a heavy reliance on animals to follow up some of these false positive tests to try and understand what's going on. So it costs more in terms of animals. And there was this publication that came out by the National Research Council of America a number of years ago called Toxicity Testing in the 21st Century. And in this, they proposed, or they challenged us to move towards a purely in vitro testing approach, discarding animal testing completely. And we have taken that challenge in our research group in Swansea, as in many other places, but we're a long way from removing animal testing at the moment. But it is important that we try to do this, because animal tests are flawed scientifically. So the results from a rodent test don't reflect human physiology or human biology. They're biochemically different and they're anatomically different. And so animal data is not always that useful, and in fact can be misleading. It also costs a huge amount of money to carry out these tests. And in Europe, there's a huge ethical concern. Public opinion has really turned against the use of animals in testing. So the last part of my talk, I'm just going to focus on a recent development of within my research group. So we've been asking the question that these DNA damage tests that we've been developing in vitro, we've been asking, can they be useful in human mutation testing? And can they specifically tell us if people have got a higher risk of having or developing cancer compared to people who don't have or won't develop cancer? And so the rationale behind this is that cancer, as we know, is a disease of mutations. And so those people with the highest levels of mutations in their bodies, in their blood or in their tissues, are more likely to be developing cancer faster than those with low levels of mutations. And so the question is, can we assess this and use it as some sort of diagnostic test? So we're particularly focused on blood cell mutations because they're minimally invasive, easy to get a blood sample, much more difficult to get tissue samples from certain patients, from certain tissues in particular. And so we are using blood-based approaches to see if the mutation levels in blood are informative in terms of someone's cancer risk. And ideally, this kind of test needs to be pretty rapid, but also needs to be reliable and robust and relatively cheap. We don't want, the National Health Service in the UK is cash limited. and it won't be able to afford a really expensive test rolled out at a population level. So I've been working for 20 years on a form of cancer called esophageal adenocarcinoma. This is a type of cancer which is started with reflux disease or heartburn. This causes a precancerous condition called Barrett's esophagus to form in the lower esophagus. And about 10% of these people who have Barrett's esophagus will get adenocarcinoma. And adenocarcinoma of the esophagus has an atrocious survival rate. Only 15% of those patients survive 5 years. It is in the bottom 3 in terms of survival rates in the UK at least. And so urgently we need more effective testing to identify these cancer patients early. And so we've been looking at these very same micronuclei that I mentioned in lymphocytes cultured from patients and asking questions, do people who have cancer have more of these micronuclei than people who are healthier? And the answer to that is yes. So as we move from, in the graph, from healthy volunteers through people with reflux disease, through Barrett's esophagus to adenocarcinoma, you see significant increased levels of these circulating micronucleated lymphocytes. And so this perhaps is a very good marker of separating a cancer patient from a non-cancer patient. And you could imagine that the level of these lymphocyte micronuclei might be informative about whether someone is a high risk of falling into this group, i.e. having cancer, or falling into this group, not having cancer and being a healthier population. The one problem with this approach, I'm very taken with the idea of measuring micronuclei in lymphocytes, but the one slight problem with this approach, apologies, my clock is chiming in the background, so it's just finished. The one problem with it is that it requires culturing of the lymphocytes after separation, after taking a blood sample. And that culturing process means that it takes up to 72 hours before you can actually score the cells for micronuclei and count the micronucleated events. And so that time could be a bit of an issue in terms of it being useful. And so we've started using this alternative mutation method called the PIG-A mutation assay, so phosphatidylinositol glycan synthase class A, which essentially is an enzyme which is involved in catalyzing the production of these GPI anchors, glycophosphatidyl'inositol anchors. So these are sticky proteins to which surface markers are tethered to cells, so things like CD molecules, CD55, 59. And so in a normal cell, the PIG-A gene is functional. It produces GPI anchors. You get CD molecules on the surface, and you can detect those with a fluorescent antibody. A key mutation in this gene knocks out the production of these GPI anchors, and so the CD molecules can't bind to those cells. Fluorescent antibodies don't detect them, and you get negatively fluorescent cells. This means that you can detect these rare mutational events by flow cytometry, by putting millions of cells through a flow cytometer and gating them. So this is shown briefly in this slide here. Blood samples taken and stained, washed, and then run on a flow cytometer. Highly fluorescent normal cells are gated, and then the very rare mutated cells are gated and counted on a patient-by-patient sample. So we use 2 different CD molecules and a red blood cell marker to make sure that we're capturing red blood cells which have lost the ability to have GPI anchors. We published our preliminary data in a paper last year, and essentially it shows a similar data to the micronucleus data I showed you earlier, in that the cancer patients have much higher levels of these mutated red blood cells than the patients with Barrett's esophagus, reflux disease, or a healthy population. But the difference with this mutational approach is that it only takes 4 hours from blood sample to actually getting the result. And so it is a much more rapid approach, and maybe it could be useful as a diagnostic tool for cancer. But what's more interesting, I think, is when we have looked at the data in more detail, we have found that when we look at the non-cancer patients, so these are patients who don't have cancer, and we've tried to assess what aspects of their lifestyle and other exposures might contribute to their mutational status, we found some really fascinating associations. And so age is a key factor. So as you age, you get more of these PIG-A mutated red blood cells. This would be expected because as we age, we accumulate more damage and our DNA repair works less efficiently. And so this was what you might expect. There is an association with obesity and overweight, with these patients having higher levels of mutation compared to the normal weight, or the people with lower weight. And this is interesting because obesity is linked to several different types of cancer. So smoking causes an increase in these mutations. We would expect that because cigarette smoke contains thousands of carcinogens which get circulated around the body. And what was unexpected and was quite interesting is the fact that aspirin reduced the level of these mutations. And so many of you will know that chemo prevention with low dose aspirin has been shown to be protective against various different types of cancer, including last year a publication showing that aspirin reduced the cancer incidence in the esophagus of patients who have this Barrett's esophagus pre-malignant condition. And so clearly aspirin is having a root, is affecting the root cause of the DNA mutations that are induced to drive cancer in the first place. And so this has led to our interest in perhaps using these approaches to assess exposures that people have and what that means for their accumulation of mutations and their future cancer risk. And as part of that, we've had some correspondence. So I was lucky enough to meet the chairperson for this morning session, Dr. Radha Saraswati. We met at a Environmental Mutagen Society of India conference in Lucknow in February, just before lockdown. And coincidentally, she's based at VIT. And we've been discussing for the last couple of months a collaborative project involving using these kind of approaches to look at different exposed populations. So so there's a slide here that my colleagues asked me to put in about opportunities at the medical school. I believe that these slides will be shared, and so people can look at these later if they want to. There's some resources, websites and things people can look at. And I will finish there and say diolch yn fawr, which in Welsh means thank you very much.
Hello.
Hello.
Yeah, thank you very much, Gareth, for the interesting and very informative talk. First of all, I would like to congratulate you about the Swansea University celebrating centenary year 2020. And there are some questions the participants have posed. Shall we take it up?
Yeah.
Okay. The very first question is, how effective are glyco-immunological treatments effective for cancer as compared to chemotherapy? And what are their side effects?
Okay, so I'm not an oncologist, and so cancer therapies is not my area of interest. So glyco-immunotherapies, I am not sure how effective they are in terms of chemotherapy, but I think that the immunologic approaches are much more specific and targeted to the individual's cancer. So they're probably going to prove to be more effective. Chemotherapy, as we know, is just a way of poisoning cells, and it's been around since the 1940s, and carries with it a huge amount of comorbidity, because it damages both healthy tissue and kills cancer cells. And so the immunotherapies are likely to be much more effective, and particularly in combination with some chemotherapies. But I must confess that I'm not an oncologist, so this isn't really my field.
Thank you. The next question is finding micronuclei a common thing, or it occurs in some specific cells?
No, I think it is very common. So, well, I say common, but in normal healthy individuals, when we've looked at blood samples from healthy individuals, you find micronuclei in maybe between 0.2 and 0.4% of cells. So 2 to 4 in 1,000 lymphocytes carry these micronuclei. So I think they are very common. They occur in a normal physiological background level. They're caused by sometimes spontaneous events, but in our bodies we have systems which look out for these micronucleated cells. So like our spleen is involved in this, and they remove some of these micronucleated cells. So I think in all tissues that have been studied, there are low-level common levels of micronuclei in those cells. So it's completely normal physiologically.
The next question, what types of cancers can be identified using blood cell mutations?
Okay, so we have focused largely on esophageal cancer because that is the type of cancer that I've had most interest in. But we have done some work with a few other cancer types, so pancreatic cancer and colon cancer, and we see similar responses there, that the cancer patients have higher levels of these mutations. And there are publications on micronucleus levels in lymphocytes in cancer patients in other cancers, which have shown that cancer patients have higher levels of micronuclei. And of course, so we use these two mutation methods because they're relatively, we're very familiar with them because we use them in other purposes, and we think they are low cost and are useful for rolling out to the population sort of level. But there's a huge movement at the moment to use genomics to actually sequence the DNA either in circulating tumor cells or in cell-free DNA circulating around in the plasma to identify mutations which are being shed from the tumors present in our bodies. So mutations can be detected for any type of cancer, probably in the blood. It probably just depends on what type of method you're going to use and the budget, which method is the most suitable. But I suspect all cancers might be detectable in inverted commas with a blood-based mutation approach.
Yeah, there's a question on what is the minimum threshold of mutations to be identifiable in blood?
So the minimum threshold, so in terms of the micronucleus assay with lymphocytes, the minimum threshold is one mutated cell, one micronucleated cell, because you detect, well, I suppose you can detect individual cells which have micronuclei, and you can detect individual cells which have a PIG-A mutation. So in the micronucleus assay, you would score thousands of cells and count individual cells which carry a micronucleus. And in the PIG-A assay, you count millions of cells with a flow cytometer, and you count individual cells which are mutated. The discrimination that you need to apply is how you separate a cancer patient from a non-cancer patient. And so that comes down to whether having, I said the background level of micronuclei in lymphocytes is around 0.2 to 0.4%. In cancer patients, it can be between 1 and 2%. And so the difference between a non-cancer patient and a cancer patient will be about 0.6 to 0.8%, which is 6 or 8 per 1,000. And so it's a small number of cells which will be the difference in a diagnostic sort of setting. But that will be set by statistical approaches to separate out cancer patients from non-cancer patients.
There's a question on checkpoints available in cell cycle. Still DNA is mutated, which lead to cancer. Can you explain?
Yeah, so that's a really interesting question. So I teach cell cycle checkpoints to genetics students. In fact, I've been marking exams for the last couple of weeks. So the checkpoints are really important in preventing cells carrying lots of DNA damage from progressing through cell division, but it's not perfect. That they miss lots of types of DNA damage, and DNA gets misreplicated. So if you have a DNA adduct, a chemically modified DNA base, and during synthesis polymerase miscopies that, that probably won't be picked up unless the polymerase stalls and triggers some sort of DNA repair. So the checkpoints will detect gross levels of DNA damage, but they will miss lots of subtle changes in DNA, and we have spontaneous mutation occurring all the time. It's estimated that something like 1 in 10 million divisions results in 1 in 10 million bases replicated gets mutated. So the 10 to the minus 7 mutational events happen every single time we divide our cells. So it's not perfect, I think, cell cycle checkpoint.
Okay, I think I'm just trying to consolidate all the questions and putting it in so that it's others could. There's a lot of questions here. So this one: is there any diversity of this biomarkers with regards to gender, age group, and ethnicity?
So that's a really good question. So I showed in the data for the PIG-A mutation data, there is a diversity in terms of age. As you age, you get more of these mutations. In terms of gender, there isn't a lot of difference with the PIG-A mutation data. However, for micronucleus data, there is a difference with females having a higher level of micronucleated lymphocytes than males. This is a known phenomenon. It's likely because of the loss of a whole X chromosome. The imprinted non-functional X chromosome often gets lost as in an aneuploidy kind of event. You lose that chromosome, has no consequence to the cell, but means that females have higher levels of micronucleated lymphocytes. And your question about ethnicity, nobody knows, because there has been, so in terms of the PIG-A mutation data, we've published data in our lab on a UK population. There's been 1 or 2 papers out of the States, and there's been 1 paper or 2 papers out of China as well. But there haven't been enough papers to really answer that question, but it would be a fascinating thing to look into.
Yeah. How is the genotoxicity more targeted in brain tumor, especially medulloblastoma? Any suggestions?
How is genotoxicity more targeted? I don't know is the honest answer. I think in some forms of brain cancer, because the cell division rate is a little bit lower, there's a suggestion that because lots of the mutations occur when cells divide, and so rapidly dividing tissue tends to have higher levels of mutation. The DNA damage can still sit there, but it doesn't get fixed as a mutational, heritable mutation if the cells aren't dividing. So there may be differences in brain cancer because of cell division rates, but I don't know the answer to that, sorry.
So then last question is, how effective is the micronuclein HCV treatment?
Is the micronucleus
in HCV treatment?
In hex?
Yeah, HCV.
HCV?
Yeah, treatment. I think it's just a random question.
I don't know what HCV.
Yeah, it must be some viral which they're going to ask. And probably they've asked many questions about joining at your place, and I think you've already given them the details. So and then I will like to thank you very much with thank you for such a lot of patience to answer such a lot of questions, and I think the organizers will take up other questions and probably post it to you. Thank you, Gareth. Nice meeting you, and a lot of change in you during the lockdown.
Yeah. Yes.
Yeah. Yes. Thank you so much, and welcome to BIT soon after all this COVID and everything, pandemic gets over. And see you soon, and wish you all the best, and take care.
Okay. Thanks very much. Bye.
Bye. Bye. Thanks to the organizers, and thank you for the session. Loganathan, please.
Yeah, thank you. On behalf of organizing committee, we would like to thank Professor Gareth for giving an excellent talk and introducing a Swansea University Medical School research, and also his own research group work on DNA damage. explaining the regularity requirement of genotoxicity studies. Thank you, sir. Also, we thank Professor Radha Saraswati for chairing this session. We thank all the participants. Nearly 800 participants attended this session. We shall be closing this session at this moment. Participants are requested to join again for the next session at 4 p.m. using the same link. Thank you all. Thank you very much.
Okay.
See you, Gareth. See you.
Bye.
Thanks, ma'am. Thanks, ma'am. Thank you. Bye. Bye.
Thank you.